Our final presenter of this session will be Jordan Willis, presenting SADIE with a very dangerous live demo.
Hi, everyone. Just like to introduce you to SADIE. Next to me is Troy. He also develops on SADIE. So just real quick, a history. This is SADIE right here. This is also SADIE. This is me debugging SADIE. This is my sister debugging SADIE. And here's the price of admission here. Here is SADIE watching me debug SADIE. So that is a little history of SADIE right there, and we'll tell you a little bit about our motivation. So SADIE is, well, I guess take a step back. You saw some very high-end apps here, pipelines and frameworks. So if you take a step back, that's what SADIE is, a library meant to be used to build those tools. So it's a low, mid, high-level API library for immunoinformatics. It's testable, it's reusable, it's portable, it's a Python library, there's one-step installation. We provide a customized and verified germline reference gene library. You can create new reference libraries on the fly. You can it maintains data formats consistent with AIR. Of course they do, or else I wouldn't be here. We have static type hints. If you don't know what that means, no problem. We can discuss it later. We have maintainable and readable code. We have a way we'd like to write code that's consistent and could be picked up by anyone to actually be, it's completely open source, so we'll take on anyone. But I do want to just mention we are in active development right now. About 30 days, we expect, version 1.0. And here's a list of our issues that we're trying to go through right now. Some of the major ones before we do a stable 1.0 release is we need our TCR sequences in there, and I know I lost half the room by just saying that. We want 100% code coverage. Everything is testable. We need a Windows and ARM build, and we want 100% documentation. So just I want to acknowledge before I go into that, Troy here, who does a tremendous amount of development on it, Anna, who's in industry, gives a ton of feedback, and Chris, who is not a computational biologist, is able to use SADIE. That's how we're trying to make it simple for the general audience. So let's see if we can do a live demo. My God, this is scary. So right here. Okay. Where are you? There we are. So, So hopefully the notebook is still hot. So first things first, everyone can follow along in a Colab notebook. We have, so I'm going to zoom in. So you can install everything. So the first thing I want to start off with is talk to my low-level people, the people that are developing on a very low level. They want just some models and some things about antibodies. So what I did is I went through the rearrangement repertoire and I made some validation models. Input sequence is a type of model that they have in the rearrangement schema. I can explicitly name that. Come on, shift. Sorry, guys. Shift. And I do tons of validation underneath the hood, just make sure everything is exactly AIR spec. And I can do it manually for all the different categories, models that are listed on the rearrangement website. So yeah, I'm just going through them and putting them in there manually. But you're probably like, okay, that's pretty stupid. I have to explicitly, define my models, but no, we got you covered. I can just give a single sequence like VRC01, and I can go ahead and annotate and fill in the models right from a single sequence. So I got all of my rearrangement schema here that I'm just printing out in a nice way, and it's ready to be used for whatever application you have. So, how do we annotate? We use the AIR module, and the AIR module has, it's able to annotate, nucleotide sequences SADIE AIR will annotate sequences. They'll verify the table, verify the fields, and return something called an AIR table object. And that is built on a Pandas data frame. And so if you're in Python and you're in data science, you definitely know what Pandas library is. I personally love it. It's, you know, the greatest thing since bread, not just sliced bread. That is how great it is. So I use it for everything, and I want to show you a little bit about like how to use it. So SADIE ships right off the bat with a couple reference libraries that are completely simple to use. So for instance, I want to, where are you? I'm gonna just run this plotting function. So the first thing I'm gonna do is I'm gonna take these catnap heavy and light sequences here. So catnap is a HIV database of broadly neutralizing antibodies. They're notoriously hard to annotate because they're crazy somatically mutated and have lots of other good stuff. So all I have to do is run call AIR, say, hey, this is human and my database is IMGT. I also want to use adaptable penalties, more on that later. That's a way we can sort of sample penalties till we get the right reading frame. So yeah, and this will take a minute to run on Colab. They don't exactly give you the they don't exactly give you like a gaming setup for free here, so it's not totally fast. But what I want to show you is we get data frames out. Let me see if I can add a code. It just B? No. Oh well. So add code right there. I just want to show you what one of these look like. They're of course data frames that are, you know, AIR compliant, and you can use them in your downstream applications. For instance, I want to show you, like, you know, how easy is it to look at your repertoire. You know, here's some human genes. What about alternate species? Now, I don't want to run these again because I don't want to take too much time. But for instance, I can use the macaque database from Nilla and Corey Watson. I call it a custom database, and that's macaque. And you can see I do run the catnap fastest sequences on here, and you see that they match with macaque germline genes. You can see like the S, which are hallmark of Nilla and Martin's workflow. So we can quickly do that, switch databases. But one of the coolest things we can do is actually build references on the fly. So I have a germline gene gateway, which is a web server that I actually host under my name, and I can actually, you know, query, you know, IMGT human, give me some V genes, and like this will tell you like what I'm expected to get, like all these different germline genes. All right, that's great, but what can I do with that? Well, I can actually query that just using normal API type methods, where I can get some germline genes from my query. In this case, I just wanted 3 human V segments. I didn't care what they were, and I'm turning them into a data frame here just to show you what that looks like. Now, but what can I do with that? Well, let's say I had like a brand new species, all right? And so I'm gonna have some fun with this. I'm gonna create an empty reference object here, and then I'm gonna add genes one at a time. I'm gonna add a human, I'm gonna add another V gene human. I'm gonna give us a little taste of our own medicine and actually splice in a mouse gene into a human. So I'm gonna have sort of a Frankenstein species here. And then what I can do is run this again and show you that we can actually annotate from custom databases just like that. Whatever sort of chimeric mouse model or human that's missing a certain allele can just be quickly turned into a database. So one other thing we can do is we can do numbering from AA amino acid sequences. So this is something Troy worked on developing using Hammer, and this is actually inspired by AIRR-C, which is an Oxford what is it? AIRR-C. It's a great program. We ported some of the code and using Hammer within here. And so we're able to actually run this here on some amino acid sequences. And what can you do? You can get right out a numbering table from, so you can get the numbering table right from this method right here. The alignment table, come on. Come on. Come on, come on. There you go. So right here, I've got all these antibodies and I've got them perfectly numbered into a Kabat type numbering scheme. We also have Chothia and all the other numbering schemes that have developed over the years. And so, in one line of code, I can just one hot encode, those things, and you're ready for your ML experiment, right? That's good. I don't want to, this is computationally heavy. And finally, I wanted to show you, SADIE can also do some clustering. We do agglomerative hierarchical clustering here. And what I'm doing is I am looking up the CDR1, CDR2, and CDR3. I'm doing a Levenshtein distance between them to calculate the distance. And then I get a distance frame and I can, visualize it in a cluster map. Of course, there's no built-in utility to plot within here, but since they're Pandas data frames, and almost every plotting library in Python expects a Pandas data frame, you're free just to go wild. I just wanted to say there's also high-level command apps. Maybe I don't even want to code. I just want to use the command line utility. You could just run SADIE from the command line, use the AIR module, give it a fast output, an AIR file, and the AIR files will be compliant to use in, your favorite software suite, implementation, or whatever accepts AIR format. All right, thank you.
Questions for Jordan?
I just want to say again, thank you for supporting the standards. Very nice work. I admire your bravery for the live demo.
Yeah.
Any other questions for Jordan? Thank you very much.
All right.